Michaela Kress is professor of physiology and head of the Department of Physiology in Innsbruck. She is a well-known expert in the neuroimmune response and interaction of the PAN pathways, and she is past president of the Austrian Neuroscience Association and also a board member of the Austrian Pain Society, and she will talk about non-coding RNAs, novel perspectives for clinical applications. Michaela, please.
Thank you very much, Christian, for this kind introduction. And I also would like to thank two Chris's for inviting me. I feel very much honored to be in this place here to talk to you about our new toys, so to speak, about non-coding RNAs. And I would like to introduce to you that these might be useful molecules with high potential for clinical applications in the future. First of all, I have to say that I have nothing to disclose. And I would like to start my talk with a general definition of the terms that are used in the non-coding RNA field. In general, non-coding RNA is an RNA sequence that is not translated into a protein. And these parts of our genome make up about 80% of the entire human genome. There are different types of non-coding RNAs. The long non-coding RNAs of about 200 nucleotides that are a group of non-coding RNAs that are where we are only at the beginning to understand what they do. They, for example, are important for gene silencing of the X chromosome, and there are a number of smaller, really small RNA sequences of 26 to 31 nucleotides. These are the PV interacting RNAs, and then there's no RNAs, small nuclear RNAs, and here in the lower part of this slide, the small interfering RNAs. These are double-stranded RNA sequences that have been used for many, many years in experimental settings to down-regulate the expression of certain genes because they introduce post-transcriptional gene silencing in animals and plants. And at the very end of the slide, this is the group of non-coding RNAs that I would like to focus on, the microRNAs. These are very small sequences of about 20 to 22 nucleotides. And this is just the first publication that ever has been published where the discovery of these new small expressed RNAs has been reported in Science by the group of Tom Tuschl. And since then, since the year 2001, the field has been expanded quite a lot. A couple of days ago, I searched PubMed for the term microRNA, and I found 42,500 something hits. And in 2 days, the number of publications has increased by more than 30. So the field is rapidly expanding. And just to show you some of the molecules that we are currently interested in, I brought this slide for you. These are short non-coding RNA sequences, and the structure that you might have seen before is quite typical. So it consists of parts where the RNA is double-stranded, and then these typical hairpin structures. These are so-called pre-microRNAs. And the active mature microRNA is actually only this red sequence here for the MIR125, and the red sequences for the others. Now, why are we interested in microRNAs in pain disorders? As we all know, there are several open questions that need to be resolved, in particular in the area of chronic pain mechanisms and treatments. And some of these questions I've put here. We still do not know why only about 50% of patients suffering from diabetes and diabetic neuropathy exhibit neuropathic pain. We also do not know why some patients after fractures develop CRPS and others do not. And what are the mechanisms and the differences in internal and external factors that are important for the introduction and the maintenance of chronic pain states are still unknown. Many studies in the past have addressed neuroimmune interactions in the pain pathway, and we now know that these neuroimmune communication processes take place in the entire pain system, starting from the nociceptor, the nociceptive terminals, where neuroimmune interaction can occur in the vicinity of an inflammation in target tissue, but also along the axon, the neuronal cell body in the dorsal root ganglion, the spinal dorsal horn, where the synaptic transmission might be affected, up to the pain processing areas in the brain. Neuroimmune interaction can take place. And just as an example, we know that after a peripheral nerve lesion, neuroimmune processes are activated in the spinal dorsal horn. As an example here shown in an experimental model, the SNI, the spared nerve injury model, we observe activation of microglia as indicated by this activation marker CD11B and another marker, the phospho-P38 MAP kinase, in the dorsal horn of the spinal cord. Such neuroimmune processes have been associated with the mechanisms of chronic pain. And what has been frequently observed also in patients, not only in animal models, is a disbalance of anti- and pro-inflammatory processes. So in particular, chronic pain patients, for example, CRPS patients, exhibit a decreased expression of the anti-inflammatory cytokines IL-4 and IL-10, and some patients also show increased levels of pro-inflammatory cytokines IL-6 and IL-1 beta, TNF-alpha. The question is, how do these disbalances arise? And another question resulting from this observation is, of course, what is the potential solution for the disbalance of neuroimmune communication? How can we rebalance anti- and pro-inflammatory processes, hoping to resolve chronic pain states? Now, I would like to show you some ideas about how neurons and immune cells, in particular in the central nervous system, can communicate. And of course, you know the neurons, they release neurotransmitters, neuropeptides, and other substances to transmit information to the next neuron. They also can use these substances to modulate glia cells in the CNS, but also the glial cells release substances that act back on the neurons or on other glia cells, which may be important to explain the disbalance of the neuroimmune signaling in the pain system. I would like to show you a couple of examples of important molecules that have been assessed in the generation of neuropathic and chronic pain states. And we all know that neurotransmitters, of course, are important, glutamate, acetylcholine, but also neuropeptides are intensely investigated. Cytokines and chemokines have been associated with chronic pain, also bioactive lipids, more recently LPA and S1P, and of course the classical prostaglandins are important. And we also know that even cell-to-cell or cell-to-matrix contacts might be involved in these processes, like proteoglycans and other matrix molecules have been found. And very recently, scientists have discovered a new route of cell communication in the central nervous system, and this is via microvesicles or exosomes, which contain lipids, for example S1P, cytokines like IL-1 beta, and microRNAs, for example microRNA 146A. These are released from glial cells, and they might act back on neurons, for example, or influence other glial cells in the central nervous system to introduce and to contribute to a disbalance of the processes that normally take place there. Before I would like to go a little bit more into the clinical perspectives, I would like to introduce to you the concepts of what we know about how microRNAs are generated. There are of course, generated in the nucleus, where there is a prime microRNA, and this primary microRNA form, this is digested by an enzyme which we call DROSHA, and this enzyme cleaves the prime microRNA into a pre-microRNA, which then uses a transporter molecule, exportin 5, to leave the nucleus. And in the cytosol, the pre-microRNA is further digested by another enzyme, which is called dicer. This produces the mature microRNA, which then can be wrapped into multivesicular bodies, as shown here. These can release their content. Then the microRNA itself in the cell can introduce translational repression or target mRNA degradation, depending on the degree of match between the nucleotide sequences of the microRNA and the target gene. The first clinical perspective can be attributed to the fact that microRNAs can be released from cells within exosomes, microvesicular bodies, and they can be observed in body fluids. And this might be one promising perspective for clinical use. Actually, this perspective has been already investigated in many other diseases and disorders, and also in the pain field. MicroRNAs have been assessed for their potential to serve as biomarkers. Disease-specific microRNA expression patterns may help us to improve diagnostics by finding the correct pain diagnosis in a quicker way, and they also might be good tools to select and attribute the optimum therapy to the individual patient. What do we know about microRNA expression patterns that might be useful or as biomarkers in the pain field? Now, the problem in the biomarker research is that large cohort sizes are usually required, and in particular regarding microRNA analysis, this causes big problems because the profiling costs are quite high, in particular since new knowledge suggests that the best way to do this analysis is via a non-biased sequencing approach. And then I would like to show you a couple of studies that have been performed in clinical settings regarding microRNA profiles. This is a very busy slide. I just want to show you that microRNA profiles have been assessed in fibromyalgia patients, in CRPS patients, in migraine, irritable bowel disease, and bladder pain. And as you might appreciate from this image, we can't really detect the details, is that in particular in fibromyalgia, there's quite a number of microRNAs that are downregulated, and the same is the case in CRPS patients, where more microRNAs are found downregulated rather than upregulated. And this is different in patients suffering from migraine. There we observe more microRNAs that are upregulated, and the same is also true in IBS and bladder pain. So in these three clinical settings, the most numbers of microRNAs are upregulated, which is not the case in fibromyalgia patients. So if we assume that microRNAs have a repressing effect, this might be indeed something that is useful for understanding mechanisms in fibromyalgia. If we assume that repressing microRNAs are downregulated, the system in fibromyalgia patients might be somehow changed into an overreactive mode. Since the discovery of the first microRNAs, about 2,800 microRNAs had been discovered by the beginning of this year. And one of the shortcomings of all these studies so far is that they've used assays with a restricted number of candidates on the assay or the chip that were used. And as you can see here, this was an assay with about 1,200 microRNAs and all the others even had lower numbers. Sorry for that. In order to get a full information about these candidates, potential candidates that might be useful as biomarkers, however, we need to assess all the microRNAs that are around. And to make our life even more difficult, in addition to the 2,800 human microRNAs that were already known at the beginning of this year, there was a publication coming out in February where another almost 4,000 microRNAs were discovered. So it's a huge effort to do this profiling analysis, in the consortium that I'm in, the ncRNA pain consortium, we have attempted to profile patients and were quite on a good way at the beginning of this year before we then had to start from the beginning and reanalyze all the data that we had collected so far to implement also these new microRNAs. So we're only at the beginning to exploit microRNAs as potential biomarkers, but I think this is an important clinical perspective which may be very useful for diagnosing pain and for choosing and selecting the correct and optimized treatment for each single patient in terms of patient stratification. The second perspective I would like to introduce to you now is more on a mechanistic basis. So far, we still do not understand fully the mechanisms of pain, in particular of the generation of chronic pain, which would be required to develop new targets and new therapies for sufficient and effective treatments. And here I would like to go a little bit more into detail about the microRNA actions in the cell. MicroRNAs are small nucleotide sequences which bind to their target genes in a protein complex which is called RISC, and the binding of the microRNA to the target gene will lead either to a translational repression if the match between the nucleotide sequences is not perfect, and this is usually the case in eukaryotic cells, or it will even cause target mRNA degradation if the microRNA perfectly matches the sequence, the target sequence at the target gene. This is usually not the case in eukaryotic systems. Now, how can we use this to modulate genes inside cells? We can use microRNA mimics, which have the same sequence as the endogenous microRNA, to introduce the translational repression, the decrease in gene expression. But we can also use antagonistic microRNAs with the antagonistic sequence. These will bind the endogenous microRNA and neutralize it. The same will be possible with a huger molecule, a so-called molecular sponge, which attracts the endogenous microRNA, binds many of these microRNA molecules to neutralize the microRNA, or we can use so-called target masks, which mask the binding site at the target gene to introduce an increased expression of the target gene, or at least to block the translational repression by the endogenous microRNA. And I would like to show you a first study that has shown that an individual, a single microRNA indeed may be effective to induce analgesia. And this was published by the group of Annemieke Kavelas a couple of years ago. She's now at the Houston Cancer Center, and she has studied a microRNA 124 in the SNI mouse model. The SNI nerve lesion was introduced here, and this introduces a robust drop in the mechanical von Frey thresholds, which suggests that these mice really become hypersensitive to mechanical stimuli. And this hypersensitivity was effectively reversed by the injection of microRNA 124, but not the mismatched control microRNA. The 124 in this setting also did not have any effects on the contralateral side, suggesting that it really was somehow targeting the changes that were introduced by the nerve lesion. Indeed, microRNA 124 is expressed in several cells that may contribute to the generation of neuropathic pain in the spinal cord, but also in immune cells. And one feature of microRNAs is that they can exert their effects in many, many cells, from individual neurons over circuits and synapses. Nociceptors can be affected, stem cells. The blood-brain barrier is an important target system, but also immune cells and glia cells are regulated by microRNA. And the fact that immune cells are targets for microRNA regulation has been nicely summarized in a review article by Hamona Sorek a couple of years ago. Neuroimiers, as they coined these microRNAs that interfere with neuroimmune signaling, are important, not only, as we believe, for pathological pain generation and maintenance, but also for a number of other neuropsychiatric diseases, and this is nicely summarized here. So based on these considerations, we may be able to use microRNA tools such as microRNA mimics or antagomies or microRNA sponges to introduce a rebalance of the disbalance of neuroimmune signaling that has been found in pathological pain states. And our first hint may come from a mutual interaction between microRNAs and the IL-6 signal transducer GP130. This is one of the few mechanisms that so far are known about how microRNA expression is really regulated. This is quite an enigmatic field at the moment still. But we know that GP130 via STAT3 signaling introduces the expression of certain primary microRNAs, and it also introduces the repression of certain primary microRNAs. And some of these microRNAs, for example, the microRNA 17 and the 20A, exert a negative feedback on the expression of STAT3 signaling. So there's a very complex interaction of transcription factors, receptor proteins, and microRNAs, which somehow interact with each other and regulate and dysregulate their own expression. One microRNA that came up from a profiling assay that we did in my lab was the microRNA 21, and I would like to show you one example of how this could contribute to the generation of hypersensitivity. We have generated a mouse model which lacks the GP130 receptor molecule selectively in nociceptors, and in this mouse model we also found a strongly reduced activity of phospho-STAT3. This is the control mouse, this is the knockout mouse, and this is the quantification here. And in the same mouse model we see that there's a significant difference in the expression of microRNA 21. So the control mice express higher levels than the knockout mice, and this nicely corresponds with the general hyposensitivity of this mouse, which I'm not going to show you in detail here. But we were just interested, and this is what I would like to draw your attention to, in a possible mechanism how this hyposensitivity could be explained. And one of the papers in the literature catched our attention. This was one paper suggesting that a certain microRNA can reduce and block the expression of a potassium ion channel. And in general, potassium ion channels are associated with regulation of the excitability of neurons. And we searched in our mouse model for the expression of potassium channels and found indeed there were 4 potassium channels that we could detect in our screen. And one of these potassium channels, the KCNA4, indeed has a binding site for microRNA 21. This is from a silico screen that we did with a reasonable score. And if we look at this ion channel, it is nicely upregulated in the mouse line that lacks the GP130 receptor, suggesting that indeed there might be a correlation between the GP130 receptor, the regulation of microRNA 21 expression, and the setting of neuron excitability in this mouse line. And we were quite happy to see that indeed in this mouse line also the potassium channel recordings were nicely fitting these other results. We see an increase in the potassium current, suggesting that these neurons, the nociceptors from this mouse line, indeed are hyposensitive or show a reduced excitability. And what was even nicer is that we saw an increase in the NIR21 expression after nerve lesion in the SNI model, and this could suggest that if the microRNA-21 is upregulated, it will set a brake to the expression of the potassium channel and introduce a hyper-excitability of these neurons under nerve lesion conditions. Now, there are several other studies who have addressed the effects of individual microRNAs in the pain field, and these are nicely summarized by a recent review of Kiran Bali and Rubini Kuna that was published last year. They're also partners in our consortium, and in summary, I think that microRNAs have at least two perspectives for clinical applications. First of all, if we understand the mechanisms how pain is generated and maintained, we can develop new drug targets and therapies, and we also may be able to exploit microRNAs as biomarkers to improve diagnostics, and both factors will help us hopefully to optimize therapies in the future for chronic pain patients. Of course, there are many people who are collaborating in this area of microRNA research and non-coding RNA research. This is my group here. We are collaborating with people in particular in Jerusalem on this issue, and we are partners in the non-coding RNA research consortium, which is funded by the European Commission and deals with the research of microRNAs, non-coding RNAs, in the generation of neuropathic chronic pain. I hope I could convince you that microRNAs are really interesting, not only for the researchers, but hopefully also for clinical use in the future. And thank you very much for listening.